Okay, so we have a biology paper here, which is February-March 2025, paper 4.2. So we'll go through this paper and discuss it all together. Alright, so let's start from question number 1. It says here, figure 1.1 shows the movement of particles through an epithelial cell in the small intestine. Alright, so we can see here, this is a representation of the small intestine. Alright, and here we have the membrane. We have 3 arrows to show movement along this membrane. We have this arrow Q, which requires energy. We have arrow R and arrow P. Okay, so what are the questions? The first question says, in figure 1.1, arrow P represents the diffusion of oxygen molecules. Describe what is meant by the term diffusion. Alright, so diffusion, it's the movement of molecules from a region of high concentration to a region of lower concentration. And of course, it's usually a random movement of these particles that are moving. So we can say diffusion is a random movement of particles from a region of high concentration of higher concentration to lower concentration. Okay, that is diffusion, movement of molecules or particles from a region of higher concentration to a region of lower concentration. Okay, the next part says, state the type of energy needed for diffusion. So the type of energy which is usually needed for diffusion is energy that is possessed by the particles. Alright, and this energy is known as kinetic energy. So we'll state here kinetic energy as the energy. Then B says, carbon dioxide molecules also move by diffusion. State the name of the process in human cells that produces carbon dioxide. Alright, so one of the processes that produces carbon dioxide in the human body is aerobic respiration. So we'll write here aerobic respiration. Okay, on figure 1.1, draw an arrow to show the direction of diffusion of carbon dioxide molecules. On figure 1.1, draw an arrow to show direction of diffusion of carbon dioxide molecules. Okay, so carbon dioxide molecules are actually meant to diffuse out of cells, move through the blood into the lungs where they can diffuse outside. Alright, yeah, so if that is the case, it means we'll have an arrow that is pointing outside of the lining of the epithelial cells. Okay, so we can draw the arrow maybe like this way. Right? Yeah, this can be the arrow that is representing the movement of carbon dioxide molecules. Okay? Yeah. So we go further. Alright. Number 1C says, if an arrow, figure 1.1 arrow Q represents another type of particle movement. Identify the type of particle movement represented by arrow Q. So you can go to look at arrow Q to identify what type of movement it represents. Alright, yeah. Okay, so when you look at it, we identify that arrow Q is representing movement from an area which there's low concentration of the particles to an area which there's high concentration of the particles. Now, this type of movement is usually referred to as active transport. So, we'll state the movement as active transport. Alright, and why? Because we can see the arrow there shows that there is use of energy, but apart from that, particles are moving from low concentration to high concentration. So say particles move from lower concentration to higher concentration. Alright. Okay, so that is the explanation. Okay, this says in figure 1.1, particle R moves from the lumen of the small intestine into the epithelial cell. Suggest why particle R cannot be starched. Okay, so it's moving from the lumen into the epithelium. Alright, why can't it not be starch? First of all, we have to know that starch is too large for it to move. Alright, so one reason is that starch is large to diffuse across that membrane. Alright, and also in order for it to be able to diffuse, it must dissolve in the blood. Okay, but starch is insoluble. So another reason could be that starch is insoluble. Alright, yeah, so this could be the reasons to why the particle is not starch. Figure 1.2 is a photomicrography of red onion cells. 1.3 is a photomicrography of the same red onion cells after being immersed in salt solution. Alright, so we have it before it was immersed in salt solution, and here it is after it's immersed in salt solution. Okay, using figure 1.2 and 1.3, describe and explain the differences in appearance of cells before and after immersion in salt solution. Alright, so before the cell was immersed in salt solution, as we can see here, it appears to be turgid. Alright, but after it was immersed, it appears to be flaccid or plasmolized because it has lost water. Okay, yeah, then apart from that, the cell after it was immersed, it appears darker than before it was what? It was immersed. Then again, when we look at it, we can see that the cell after it was immersed, it has somewhat detached from the cell wall. The cell membrane has detached from the cell wall, unlike before immersion where it had not yet detached. Alright, okay, so we can light those explanations over here. Appearance in that, so we'll say before immersion, the cell appears turgid. Right, that's one thing. And also before immersion, the cell membrane pushes against the cell wall. Alright, pushes against the cell wall. Okay, yeah, and of course before immersion there's increased turgor pressure, or there's high turgor pressure, which enables the cell to push against the cell wall. So again, before immersion there is increased or higher turgor pressure, and then after immersion, what is appearing? After immersion, the cell appears flaccid. The cell appears flaccid, which means it has now shrunken in one way or another. Alright, flaccid due to loss of water by osmosis. Alright, yeah. Then after that, after immersion, the cell membrane has detached from the cell wall. The cell membrane detached from the cell wall, does not push against the cell wall anymore. Alright, and of course after immersion, the cell appears to be darker. Alright, so that is what we observe after immersion. Okay, move to the next question. It says here, the internal body temperature of a person was recorded. Figure 2.1 shows the results. Okay, so here we have the graph to show how the temperature was recorded. As we can see, the internal body temperature is kind of ranging from 36 to 38 with time. Using the information in 2.1, calculate the temperature range for the internal body temperature of the person. States the unit, so it is in degrees centigrade. Alright, how are we going to identify the temperature range? We'll take the highest minus what? Minus the lowest. So the highest temperature is 38 and the lowest is 36. So we'll take 38 minus what? 36. And when we subtract the two, we'll get 2 units. Alright, so the difference is going to be 2, and with units it is in degrees centigrade. Alright, so that is the range. And then on figure 2.1, draw a line to show the set point for internal body temperature of the person. Alright, so this set point for internal body temperature of a person is usually 37. You can see the temperature is like constant on 37. It either falls below 37, whereas it is forced to rise, or changes take place so that it can rise above 36, and if it goes above 37, which is around 38, it's again supposed to fall back to what? To 37. So it's 37 is a constant, all right? It goes above 37 a little bit to 38 and falls below 37 a little bit to 36, but not more than that. Okay, so we'll draw the indication over here, all right, on 37. Okay, so draw the indication over here. All right, yeah, so that is the range for what? For temperature of the person. Okay, the maintenance of internal body temperature is an example of homeostatic control. Say the name of the mechanism for homeostatic control. All right, so homeostatic is usually controlled by a process known as negative feedback, whereas a change acts as a stimulus for what? For a change. So we'll state here negative feedback. All right, yeah. Okay. On figure 2.1, region F shows change in body temperature. Explain how the body causes the change in body temperature shown. Okay, so region F. Okay, that's region F. As we can see, it's ranging from 36 to 38. Then the question says, explain how the body causes the change in body temperature shown. All right, so the temperature is since actually 36 to 37 and to 38. So, which means if we have a temperature above 37, like 38, this is an increase in temperature. All right. So this increase in temperature acts as a stimulus. Okay. So it will be detected by the brain, and then after it is detected by the brain will command the hypothalamus, which is the temperature center, to bring about changes which include lowering the temperature. And when the temperature is at 36, again the brain will detect, all right, and command about a change which will be raising the what? The temperature. All right, so what will happen, or how will the body control this change? First of all, we'll say that the receptors detect the change in temperature, the first point. So receptors detect change in temperature. Okay, yeah, and then and transmit it to the brain. And transmit it to the brain through sensory neurons, through sensory nerve or sensory neuron. All right, then the brain brings a response, and a response is then brought to raise the temperature from 36 degrees centigrade through 1 vasoconstriction. All right, so we have a change in temperature or a fall in temperature, vasoconstriction. Yes, so vasoconstriction means narrowing of blood arterioles so as to allow less amount of blood to flow underneath the skin and hence reduce heat loss. Okay, that is the first thing. But apart so vasoconstriction, so less amount of blood flows to skin surface and less amount of heat is lost. Apart from vasoconstriction, we have shivering. Okay, so shivering this is more like a contraction of skeletal muscles, all right, so that they can generate what? Heat. So this one here, I will say it results from contraction of skeletal muscles, contraction of muscles to generate heat. Okay, yeah, this is another thing that will take place. Then apart from that, we'll have increased rate of metabolism. All right, apart from increased rate of metabolism, we'll have the hair erector muscles stand up, the hair stand up. Okay, so here I will say another thing is hair stands up, hair stands up, so as to trap the heat that is being lost. But apart from that, another thing that is going to appear here is increased metabolism. Increased metabolism so that more heat is produced within the body. All right, yeah, so those are the things that will take place. Okay, figure 2.2 shows a cross-section of human skin. All right, as we can see here, this is the cross-section of human skin with different parts of the body. State the letter of the structure shown in figure 2.2 that produces sweat. Now, sweat is usually produced in the sweat gland. Where is the sweat gland? Over here. The sweat gland is this part here. All right, so it's represented by letter A. The letter we use here is state the names of structures C and D. Let's look at structure C first. All right, so structure C is this part over here. What is the name of this structure? This one here is the hair erector muscle. And then D is this part over here. D is usually representing the sensory neuron that is scaling this particular impulse. So C is hair erector muscle. Hair erector muscle, and D is sensory neuron. Yes. Okay, so we move further to question number 3. Question number 3 says 3.1 shows the percentage of water in different structures of the human body. Now we can see the different structures of the human body. Here we have the brain 73%, blood 94% of water, kidney 79%, muscles 75%, eyes 95%, heart 75%, lung 83, bones 31. Okay, the mean mass of the human eye is 28 grams. Using the information, calculate the mass of water in the human eye. Give your answer in 2 significant figures. All right, the easiest way to identify the mass is the mass of water, right, is taking the percent and multiply it to the mean mass. All right, now the percent of the eye is 95%, so it's equivalent to saying the mean mass is 28 multiplied to 95%. All right, or you can say 28 times 95 over 100. All right, so when you work out this one here, you get 27. All right, yeah, so the mean mass, using the mean mass of human eye is 28. Mass of water, yes, the mass of water is going to be 27 grams. Mind you, we got it from taking the mean mass, multiplying it to the percent of water in the eye. All right, Roman 2 says, describe the importance of water in the human body. All right, now what are the importances of water in the human body? First of all, water acts as a solvent. Solvents dissolve substances in the body, so it is a solvent. It dissolves the various substances in the body. But also, it carries a lot of transportation roles. All right, so we can say here, it transports substances within the body. It transports substances within the body. All right, these substances can be nutrients, can be gases, can be wastes. But also it regulates temperature. All right, another role is temperature regulation. Okay, yeah, temperature regulation. But also it can be used in the process of digestion, so we can say here it's used for digestion. Okay, these are some of the importances of water in the human body. Okay, let's go to B. B says cholera is a disease caused by pathogen in contaminated water. Figure 3.2 is a diagram of cholera pathogen. As we can see it here, this is representing the cholera pathogen. State the name of the structures L and M shown in figure 3.2. Now, M is this small dot-like thing here. This one here is representing a ribosome. But L is this part here. This part here is the inner membrane, which is actually referred to as cell membrane. Alright, so L will be cell membrane and M will be ribosome. Okay. Identify two features shown in figure 3.2 that are typical of prokaryotes. Alright, so what are the features that we can see here that are commonly found in prokaryotes? One of them is plasmid, this round structure over here. So we'll say plasmid, but apart from plasmids, prokaryotes usually have cell wall. So we'll say that they have cell wall, but apart from having cell wall, they have plasmids, but also you can see that there's no nucleus around the DNA. So it's just a circular DNA found in the cytoplasm of the cell. Okay, state the type of pathogen that causes cholera. Alright, so the pathogen that is causing cholera falls into the group of bacteria. So we'll state here, bacteria. Okay. The scientific name for the pathogen that causes cholera is Vibrio cholerae. State the genus name for this pathogen. Now, again, to the concept of binomial nomenclature, we assign two names, all right? The genus name and the species name. The genus name is the one that starts, and it usually starts with a capital letter. So, Vibrio here will be the genus name. Alright. Okay, explain how the cholera pathogen causes dehydration of the human body. Alright, it causes dehydration because cholera pathogen results into production of toxin. The toxin then causes secretion of chloride ions, and then accumulation of chloride ions in the small intestine causes water to move into the small intestine by what? By osmosis, and this is due to difference in water potential. Then after that, this water which is accumulated results into diarrhea. Alright, so diarrhea is like passing of water feces. So much water is being lost and eventually resulting into dehydration. Alright, so we'll state here cholera results into production of a toxin and of a toxin. The toxin causes secretion of chloride ions in the small intestine. Chloride ions in the small intestine. The chloride ions lower the water potential. In the intestines, causing water to move into the intestine by osmosis. Resulting to diarrhea. Okay, so this is what is happening. Toxin causes chloride ions results into movement of water results into diarrhea, which causes dehydration at the end of the day. Okay, complete the sentences about the use of plasmids in genetic engineering. During genetic modification, human DNA and plasmid are cut with restriction enzymes. This creates dash which are joined together using an enzyme called dash. The modified plasmid containing human gene is also referred to dash plasmid. Okay, so during genetic modification, human DNA and plasmid DNA are usually cut by restriction enzyme. Alright, once they are cut by restriction enzyme, they form a DNA which has sticky ends. So we'll say here this creates sticky ends. which are joined together using an enzyme DNA regase. Alright, so DNA regase is the enzyme that is being used here to seal these two different DNAs. The modified plasmid containing the human gene is called, so the new DNA, which has a mix of two different other DNAs, is usually known as recombinant plasmid. So say here, recombinant plasmid. Okay, move to the next question. It says here, figure 4.1 shows part of human placenta and umbilical cord. The arrows show the direction of blood flow. So we can see the arrows. For example, we have these ones here. Okay, we have the other ones down here. All right. Describe the functions of the placenta and umbilical cord shown in figure 4.1. All right. So let's start naming them all together. Placenta is it's a part which usually produces hormone progesterone during pregnancy. All right, so we'll state here, placenta produces progesterone hormone. Why is the hormone being produced? It's being produced so as to maintain pregnancy. All right. Apart from that, they have some other shared functions. All right, because you know, the placenta is this organ of exchange here, all right? But then again, it has the umbilical cord, this part over here. Inside the umbilical cord, in here, you have umbilical vein and umbilical artery. Okay, so it's like they are connected. Okay, so what are the other functions that they do apart from producing progesterone hormone? Okay, so we say that they transfer substances from the mother to the fetus. Example, nutrients. All right, transfer substances from the mother to the fetus. Apart from doing that, the other thing is they carry substances from the fetus to the mother, like wastes. Okay, so they transfer substances from the fetus to the mother. Example, wastes. All right, yes, but also they transfer antibodies to the child. All right, and in other words, they are protecting the child throughout the pregnancy period. All right, yeah, so we can say here, transfer antibodies or pass immunity to the child. All right, to the fetus. Antibodies to the fetus, so they are passing immunity to the fetus. Okay, Roman 2 says a fetus develops inside an amniotic sac. Describe the functions of amniotic sac and amniotic fluid. Okay, so what are the functions of amniotic sac and amniotic fluid? First of all, it's to protect the fetus from mechanical damage, and then it is to nourish the fetus, and then again it is to allow movement for what? For the fetus. All right, but also to regulate temperature. But then again, amniotic fluid is usually found inside amniotic sac. So the sac is usually producing what? Amniotic fluid. So say here, amniotic sac produces amniotic fluid. Amniotic fluid. But then again, we can say another function is to prevent mechanical damage. Prevent mechanical damage of the fetus. All right, apart from preventing mechanical damage from the fetus, allows movement of the fetus. All right, allow movement of the fetus, but also the fluid helps to regulate temperature. So here, regulates temperature. All right, so these are some of the functions of amniotic fluid and amniotic sac. Syphilis is a sexually transmitted infection, STI, that can be passed from a mother to her fetus. State the name of one other STI that can be passed from mother to fetus. All right. Another STI that can be passed is AIDS. Okay. So we can say here HIV AIDS. Okay. That's another STI. State 2 ways to control spread of STI. One way to control spread of diseases is through educating or education. All right. When people are educated, they can be aware that this is not correct. But also we can use antibiotics in case people have been discovered or tested positive for the disease. And also we can do abstinence. All right. Abstinence. Okay, if people are not sexual active, they can avoid getting STIs. So it's one way of preventing spread of STIs. Okay, question number 5 here says 5.1 shows a cross section of a leaf. So this is a cross section of a leaf. In figure 5.1, draw a circle around the vascular bundle. So the vascular bundle is usually found just around or within the sponge mesophyll cells. All right, and it will have a concentration of phloem and xylem vessels. Okay, so if we are to identify the vascular bundle, it can be this part, can be this part. All right, it can be these parts. Alright, so those there can stand as vascular bundle. Okay, on figure 5.1, draw a label line and the letter X to identify the parasite mesophyll tissue. Alright, so the parasite mesophyll tissues are usually just after the epidermis, after the upper epidermis. Alright, so if you are to identify the parasite mesophyll cells, there should be these ones here. This here an arrow. Yes, so these ones here are the parasite mesophyll tissues, X. Okay, state the name of the cell structure in parasite mesophyll cells where photosynthesis occurs. So in parasite mesophyll cells, we do have chloroplasts, which can trap sunlight. Now, this ability of them trapping sunlight allows for photosynthesis to take place in them. All right, so we will name here chloroplasts. Okay, describe the functions of the tissues in vascular bundle in a leaf. Okay, what is the function of the vascular bundle? All right, so we have functions of xylem, we have functions of phloem. Phloem usually transports assimilates, or we can say the products of photosynthesis like sucrose. While xylem, this one here, transports water. All right, but apart from transporting water, xylem usually provides mechanical support. All right, yes, so you can say here xylem provides mechanical support for the leaf. Apart from that, xylem transports water, and then phloem transports sucrose. And other nutrients like amino acid from the process of photosynthesis. B says the leaf shown in figure 5.1 is from an aquatic plant adapted to live in water. The leaves float on the surface of water. State the term used to describe plants that are adapted to live in water. Alright, so the plants that are adapted to live in water are usually known as hydrophytes. Alright, so we'll write here hydrophyte. Identify feature T shown in 5.1 and explain how this feature adapts the leaf to float on the surface of the leaf. Where is feature T? Alright, so this part here is what feature T is, and it's just like a spherical part. Okay, so this spherical part here is a feature that represents air spaces. Alright, so say here air spaces. Okay, so how does it make the leaf adapted to floating in water? Air space is like an empty space, so it reduces what? The density allowing the leaf to float in water. So here we'll say it decreases the density of the leaf, allowing it to float on water. Okay, explain one other adaptation of this group of aquatic plants. Okay, apart from having maybe lots of air spaces, these plants usually have very little cuticle, or sometimes they don't have cuticle at all. Why? Because they do not need to prevent rate of transpiration. Okay, but apart from that, they also have stomata on the upper surface of the leaf. Why? To allow gas exchange. So we'll say here they have only on the upper surface. They have stomata on upper surface. They have stomata on upper surface to allow gas exchange. Yes, so that's one other adaptation, but also they have thin cuticle on them. All right. Question number 6 says microplastics are pieces of plastic with diameter less than 0.5 centimeters. Figure 6.1 shows the mass of microplastics in the ocean between 2000 and 2040. The data between 2000 and 2020 is an estimate. The data from 2020 is a prediction. Okay, so you have the mass of microplastics in million tons from the year 2000 to the year 2040. Okay, suggest why mass of microplastics between 2000 and 2020 shown in figure 6.1 is an estimate. All right. Okay, why do we estimate this mass between this area? All right, so first of all is because not all microplastics are usually collected. Okay, the mass that they might have measured or estimated is just a little portion or a certain amount of microplastics, some of which have not been collected. All right, so if that is the case, it means we are just estimating. We have not taken the actual mass. Okay, maybe we have not come across it. Okay, then again, the microplastics are produced in very large quantities in different places. So the mass that will be identified or will be used is just a little amount of what is present. All right, some of which is not identified, so it is not actually measured. Okay, and of course we are speaking about some microplastics that can be consumed. Maybe they are inside the organism in an ecosystem or in a food web. So those ones there are not measured. Okay, it's only certain samples that are identified. Okay, so why is mass of microplastic assumed as an estimate? First of all, because not all microplastics are measured. Okay, that's the first thing. Not all microplastics are measured. All right, but from that, only samples were taken. Only samples were taken or described. So the mass that is measured is only for certain samples. Were described and measured. Okay. Using figure 6.1, calculate the predicted percentage increase in mass of microplastics in the ocean between the year 2000 and the year 2040. Calculate predicted percentage increase in mass between 2000 and 2040. Let's read the reading in the year 2000. Okay, in the year 2000, it's here. Okay, so what is the estimated population? In mass of microplastics? If this is 0 and this is 0.5, then we can assume that this here is like, let's say 0.5, 0.2, 0.3, 0.4, 0.5. All right. So from the scale, in the year 2000, it has to be 0.2 million tons. Right. So in the year 2000, the reading was 0.2 million tons. Okay. But what about the year 2040? How will it be? Let's look at 2040. 2040 is here. All right. So this is 1.0. This is 1.5. Now 1.5. This should be 1.6. 1.6, 1.7, yeah. So the year 2040, the reading is 1.7 million tons. All right. So to get percentage increase, we'll take the reading from 2040 and subtract the reading from the year 2000. So I'll take 1.7 minus 0.2 million tons, divide it to 0.2 million tons. This whole thing, we multiply it to 100, but we know that it is million tons. All right. Okay, so 1.7 minus 0.5, this is minus 0.2, this is 1.5. 1.5 divided to 0.2 times 1000. All right, we get approximately 750%. All right, so the percent here is going to be 750%. Okay. B says phytoplanktons are producers found in the ocean. Phytoplankton absorb microplastic into their cells. Describe what is meant by the term producers. So producers, these are the organisms that make their own food. Okay? How does it make their own food? It makes its own food by the process of photosynthesis. So we say that microorganisms, these are organisms that make their own food by the process of photosynthesis. All right, so that is what it means when we speak of producers. Figure 6.2 shows a shearwater bird. Shearwater birds feed on fish in the ocean, as we can see it is flying above the water. Suggest how microplastics can end up in consumers such as shearwater birds. Okay. Now the thing is, these organisms here are feeding on what? On fish. All right. And microplastics are so small. Apart from being so small, they don't decompose. Okay, they don't break down. So they can be deposited or thrown into the water. All right, now because they are so tiny and non-biodegradable, you find that they are eaten by what? By fish. Okay, so once they are eaten by fish, they will remain into the fish's body. And then the shearbird will come and eat that particular fish. It will eat the fish with the microplastics that are within it. Okay, so say here, microplastics are non-biodegradable, meaning that they cannot be broken down. All right, they can be consumed by fish. Okay, they remain in the fish, and the shearbird feeds on the fish with microplastics. Okay, so when it feeds on the fish with the microplastics, it ends up getting them also. Okay, the population of shearwater birds is decreasing. Describe 3 ways the population of shearwater birds can be conserved. All right, so what are the ways that we can do in order to maintain this species? First of all is doing captive breeding. It means we capture these birds and allow them to breed, so we have a lot of them of such kind. Apart from that, we can also say that providing education and also monitor hunting and poaching. All right, yes, and also establishing laws to maintain these organisms, and also protecting especially the habitats in which they live. So here we can say one way is providing education on importance of monitoring these species, but also we can do captive breeding, capture them and allow breeding so that they can have more species of such kind. But also we can have establishment of laws for poaching and hunting, establishing laws of hunting or for hunting, all right, so as to conserve the species and prevent poaching. Okay, explain the risks to a population if its population size decreases. Now, what happens when a population size decreases? We will have something known as inbreeding, meaning that species that are closely related, they keep on breeding, so we will not have genetic variation due to crossbreeding of species. All right, so one thing is inbreeding. All right, this is one of the effects when the population decreases. But apart from that, when we have inbreeding, it means we have reduction of genetic variation. So here, reduction of genetic variation. Apart from reduction of genetic variation, we can have extinction. Okay, because the species don't breed that much, and if they breed individuals that kind of look alike. We don't expect them to have such a good adaptation to the environment, and of course we can expect them to run extinct. All right, yeah, all right, and that was the last question for this exam. Thank you.